Today's topic is History of Clinical Trials. Clinical trials are a set of tests in medical research and drug development that generate safety and efficacy data, that is information about adverse drug reaction and adverse effect of other treatment for health intervention, for example drugs, diagnostics, devices, therapy protocols. History of clinical trial before year 1750 where Persian physician and philosopher Avicenna in the canon of medicine he laid down rules for the experimental use and testing of drugs and wrote a precise guide for practical experimentation in the process of discovering and proving the effectiveness of medical drugs and substance. He laid out the following rules and principles for testing the effectiveness of new drug and medications. Earlier rules are the drug must be free from any extraneous accidental quality. It must be used on a simple, not a composite disease. A drug must be tested with two contrary types of disease because sometimes a drug cure one disease by its essential quality and another by its accidental ones. The quality of the drug must correspond to the strength of the disease. The time of action must be observed so that effect and accident are not confused. The effect of the drug must be seen to occur constantly or in many cases, for if this did not happen, it was an accidental effect. The experimentation must be done with the human body. For testing in a drug on the lion or a horse might not prove anything about its effect on men. Perhaps the first ever clinical trial was James Lind's demonstration in 1953 that citrus fruits cured scurvy. He compared the effects of various different acidic substances ranging from vinegar to cider on groups of sailors and found that the groups who were given orange and lemons had largely recovered from scurvy after 6 days. Before one can initiate testing in human beings, they must conduct extensive preclinical or laboratory research. Research unusually involves years of experiment in animal and human cells. If this stage of testing is successful, the sponsor then provides this data to the FDA requesting approval to being testing in humans. This is called an investigational new drug application, that is IND. If approved by the FDA, testing in humans begins. This is done through a formally written and approved protocol. A study plan on which all clinical trials are based are protocol, carefully designed to protect the health of participants, describes what type of people may participate in the trial, gives detailed schedule of test, procedures, medication, dosage and length of the study. Principal investigator is responsible for assuring that the protocol is strictly followed for each participant. Randomization of trials. Research assigned patient by chance to either a group taking the new diagnostic or treatment against, similar to flipping a coin. Randomization helps avoid bias. The assigned group are often referred to as arms. Example, patient #1 is assigned to arm A of the trial, which involves the new modality or treatment. Patient 2 is assigned to arm B, which is the standard modality of treatment.